Okay, AP Biology students, this is the ending of our lecture on enzymes and metabolism. It's good to be back in business here. So what we see here, we finished with the induced fit model, which basically what you could see is the substrate is going to enter the enzyme at the active site, and the enzyme will fold itself around the substrate there, and that's where we get that induced fit. So the enzyme fits snugly around the substrate, kind of like a clasping handshake. And we also spoke about how an enzyme's activity can be affected by temperature, pH, and other chemicals in the body or introduced into the living system. So you could see different examples. Here we have in part A, the rate of the reaction, temperature, and what would happen there. And then down below in part B, you have the optimal ranges for pH for two different enzymes, one being an intestinal enzyme, the other being a stomach enzyme. Cofactors are non-protein enzyme helpers, such as minerals. So here's where you can see chemicals playing a role with enzyme activity. Non-protein based helpers, such as zinc, iron, and copper, would be chemical factors. You also have coenzymes. These are organic cofactors. Molecules like vitamins will help in aiding the functioning of enzymes. It could be either bind to the, most likely the substrate, so it's able to go into the active site of the enzyme and promote enzyme catalysis. Enzyme inhibitors. Enzyme inhibition is going to prevent the enzyme from doing its job. There are two types of inhibition. There's the competitive inhibition or competitive inhibitor, and there's non-competitive inhibition or a non-competitive inhibitor. In competitive inhibition, another substance is going to compete with the substrate and bind to the active site. In non-competitive inhibition, it's going to bind to another part of the enzyme. And ultimately, when that substance binds to another part of the enzyme, it's going to cause a change in shape in the enzyme. So you're going to see a conformational change within the enzyme and ultimately the active site being changed will make it non-functional. When something binds to the enzyme at an area other than the enzyme itself, the enzyme's active site, we would call that its allosteric site. And you could recall that from the lesson. So yeah, the active site of the enzyme and anywhere else where something's going to bind that change the shape of that enzyme is the allosteric site, ALLOSTERIC. So here you can see examples of both competitive and non-competitive inhibition. So here you can see the normal binding. Here's a competitor binding to the active site there, not allowing the substrate to come in. And here you have a non-competitive inhibitor binding to the enzyme, changing the conformational structure of that enzyme. That there would be the allosteric site. So regulation of enzyme activity. Why do we have inhibition? Ultimately, if we start to produce a lot of a product, ultimately the product can come back in and bind to the enzyme and say, all right, we have enough of this particular end product. We don't need any more. We could stop producing it. It's all part of that negative food feedback loop mechanism where you want to keep things within balance, within normal range. So regulating enzyme activity, to regulate metabolic pathways, the cell can switch on and off genes. We're going to look at this in greater detail when we get into molecular genetics. It will switch on and off genes that are going to encode specific enzymes. Allosteric regulation, protein functions at one site is affected by the binding of a regulatory molecule at a separate site. So that's the non-competitive inhibition. That would be the allosteric site. Often that is the end product of a metabolic pathway that might bind to say, okay, we have too much of what we need here. So when you have allosteric regulation, you could have an activator. Activators are going to stabilize the active site. Inhibitors are going to stabilize the inactive form of the enzyme. And then you could have cooperativity. And basically, that's going to occur when one substrate is going to trigger a shape change in another active site and therefore cause an increase in catalytic activity. So here you can see allosteric activators and inhibitors, active form versus inactive form. Allosteric enzyme with 4 subunits. You have an active site, which is 1 of 4, and you have the regulatory site, which is 1 of 4. So there you can see an activator coming in. It's going to stabilize the active form, or you could see an inhibitor coming in, and ultimately it's going to stabilize the inactive form and turning those enzymes on or off as they would function in a particular metabolic pathway. Here you can see another type of allosteric activation. This is cooperativity. Inactive form, substrate will come in, it will bind and stabilize the active form of that enzyme, therefore increasing that enzyme's catalytic rate because it's going to stabilize that active site and it will allow for other substrates to come in and bind to the enzyme to be converted to a product. Ultimately, as I said previously, this is all part of feedback inhibition. The end product of a metabolic pathway shuts down the pathway by binding to the allosteric site of an enzyme. This is going to prevent wasting chemical resources, and it's also going to increase the efficiency of the cell. So you can see feedback inhibition. Here we have the active site of enzyme 1. It's no longer able to catalyze the conversion of threonine to an intermediate A. The pathway is going to be switched off. So here you have isoleucine. Isoleucine, remember, and threonine are amino acids that are going to bind to the allosteric site of this enzyme. Isoleucine is going to be used up by the cell. Ultimately, this will create the active site. When that's used up, it's going to activate that enzyme, and the initial substrate there, threonine, is now able to come in and bind to the active site of the enzyme. So enzyme 1 is threonine deaminase, and that's going to ultimately come in. You'll form intermediate molecule A. An enzyme will come in to form intermediate molecule B. Enzyme 3 will come in to form intermediate molecule C. Enzyme 4 will come in to catalyze the conversion of intermediate molecule C to intermediate molecule D. And then enzyme 5 will catalyze intermediate molecule D to be converted to the end product, which is isoleucine. Once again, when we have enough of that product available, then we have isoleucine binding to the enzyme itself at the allosteric site to change the conformation of the active site and pretty much say, all right, we have too much isoleucine produced now. We could stop until it's used up, and it's all part of that homeostatic negative feedback loop inhibition. That's basically it on our chapters on enzymes. I hope you enjoyed the lecture. I knew there wasn't much more to do. From here, we're going to go with metabolism into our 2 metabolic pathways, photosynthesis and cellular respiration. Thank you, everyone, and have a nice day.